I'm very excited to be back to get a chance to update you on Avrobio. We made a lot of progress since I presented here this time last year. We have a lot of data to share, so it will be a race against time. And I should begin by framing the company. We are an ex vivo lenti gene therapy company that targets multiple serious rare genetic diseases, lysosomal storage disorders. The goal of the company is to free tens of thousands of these patients from a lifetime of disease, to really halt disease progression, and to untether these patients from today's standard of care, which are biweekly infusions every 2 weeks for the rest of their lives. So that is what we've set out to do. I just quickly say that I will be making forward-looking statements based on our current understanding of future events as of today, and statements are subject to risks and uncertainties, so I point you to our 10Q. Quick point on the team. I think we're really proud of the people that we've been able to attract to Avrobio, and it's really been a conscious co-culture of two skill sets, of real lifetime pioneers in the cell and gene therapy field, people that have really built and led some of the early pioneering companies, CMOs, and really academic leaders in this field over the last 10 to 20 years, but also to really bring into the leadership team and all the way through the company people with that rare disease phenotype, and people that have really had an impact in companies like Genzyme and Shire and Alexion and so forth. And we really think a critical success factor is to really mesh these two skill sets to be able to effectively build a gene therapy company targeting rare disease. You've seen more than one permutation of an ex vivo lenti gene therapy diagram, so I'll just make a quick point that all of our disease targets leverage one platform, so one cell and gene platform where, of course, it's autologous. We collect the patient's blood, we select out the CD34 hematopoietic stem cells, we then give them an upgrade with our proprietary 4-plasmid lentiviral vector system to deliver the healthy gene. The modified cells are cryopreserved, where they're then thawed and infused in a straightforward one-time procedure. Because it's one platform across the company, it's afforded us the ability to invest a lot of energy and a lot of time, effort, resources to optimize it. And towards the end of the presentation, I'll talk about where we are in that optimization work. But first of all, our pipeline, Fabry, Gaucher, cystinosis, and Pompe. So there is an incredible congruency to the portfolio. These are all lysosomal storage disorders. And now standing here today, really being able to say that we have demonstrated pretty compelling proof of concept in Fabry disease, we think that that halo does actually alter the probability of success in the sister indications, Gaucher, cystinosis, and Pompe. So I'll share a lot of our Fabry data with you. We're very excited that we are now actively enrolling, having achieved IND for Gaucher and cystinosis. So if you do invite me back next year, hopefully I'll be able to share some data on both of those two additional indications. Now, in rare disease, sometimes you think small. I'll just make a few points about these diseases collectively. First of all, they comprise collectively about 30,000 patients, and the Shires and Genzymes, or Takedas and Sanofi, sell about $4 billion annually in these diseases. So they do represent very significant commercial potential, and also they cost the healthcare system a fortune. The blue graphs are annual costs, but there is an Erasmus publication which suggests that the lifetime cost of a Fabry patient on today's standard of care is $14 million. And as you can see, it could be higher in some of the other diseases. So very expensive, commercially attractive markets. So then you may ask, if they're so well served, why even contemplate a gene therapy? And it really comes down to pharmacokinetics. Each of these graphs, these spikes, represent a patient visiting an infusion center every 2 weeks forever, for life. And so, of course, you think of cost, compliance, quality of life, but the issue centers around the kinetics. And said plainly, it's the right enzyme, bad pharmacokinetics. The proof source is if you test a patient's blood 12 hours after one of these biweekly infusions, the enzyme's gone. So a minority may be taken up intracellularly, but the majority is processed by the liver and wasted. And so that really is the central hypothesis of gene therapy and of Avrobio is what if you could deliver 24/7 expression of enzyme, distributed, essentially bathing cells, tissues, organs. Hopefully you would not just slow down disease, which is what ERT, enzyme replacement, does, but halt disease progression. And that's what we've set out to do. So a quick mechanistic point is these modified cells are infused, migrate to the bone marrow. A minority of them will engraft for the life of the patient, called the long-term engrafting cells. And the mechanism comes from their progeny, their daughter cells, which form the blood lineage, the leukocytes. And that these cells, billions of these cells, manufacture, but importantly also distribute active protein. So here you see a kidney, and you would anticipate that cells like DCs or macrophage would visit the kidney, a site of inflammation. And so in that sense, actually, it's a delivery system, not just spewing out enzymes systemically. And this really is a differentiating feature of the ex vivo lenti approach. So to pivot to the Fabry data, we have a phase 1 trial that is fully recruited, 5 patients, and we're 3 patients into a global phase 2 trial. And I don't have time to share all the data, but I'll share a few snapshots. One very important patient data to share is from our global phase 2 trial, which are ERT-naive patients. And this was a 21-year-old classic male Fabry Australian patient looking at his baseline of the average number of GB3, which is the substrate, inclusions per peritubular capillary at baseline versus 12 months. This is very important for us because it's our primary endpoint, and it is the approvable endpoint for a company called Amicus with a chaperone therapy. So we do think that from a regulatory point of view, this data is quite meaningful, but also mechanistically it's meaningful because it demonstrates the ability of this gene therapy to actually impact tissue. And so, of course, the result is compelling, an 87% reduction. And our global PI, Professor Mark Thomas, who's a nephrologist at the Royal Perth in Australia, said that the reduction is either at or darn close to full effective clearance. So we're extremely encouraged by this. And this functional data is also supported by a number of biomarkers. I don't have time to go through all of them, but some of them that are very meaningful in the field of Fabry disease are a reduction in the plasma of the toxic metabolite, lyso-GB3. And here you see an 87% reduction of this biomarker, and the substrate itself in the plasma, GB3, showing a 73% reduction, which is down to the normal reference range. So we're extremely pleased with what we've seen so far in our phase 2 trial. To jump to some of the longer-term data from phase 1, this is a different patient population. This is a patient that for 10 years received biweekly infusions of enzyme replacement therapy, had his enzyme replacement therapy withdrawn for a period of time, was reinitiated, so received 2 medicines, ERT and gene therapy, and then for approaching a year is now on gene therapy alone. And so you have this wonderful within-patient control where you can look at how effective in reducing lyso-GB3 was 10 years of biweekly infusions. And what you see is that on gene therapy alone, there's a further reduction from the ERT baseline of 41%, and a sustained reduction going out on this slide, 28 months. So this is patient number 1. What's also emerging is a picture of reproducibility. Across the first 4 patients in the phase 1 trial, we're seeing a consistent further reduction of lyso-GB3, the toxic metabolite, versus the patient's ERT baseline. So we're extremely satisfied with what we're seeing, and we'll, of course, continue to track these patients. But looking at the big picture, to be able to outperform the baseline ERT is really what we set out to do with this gene therapy. And then the final important question, of course, for one-and-done therapies in general is durability. And one important thing to understand about the mechanism of ex vivo lenti gene therapy is we collect an awful lot of CD34 hematopoietic stem cells, transduce all of them, and infuse all of them back in, knowing 99.9% of them are progenitor cells, are what's called short-term engrafting cells. And so what you get is this bolus of activity in year 1, which is great, which really helps the patient. but the long-term durability actually comes from a smaller population of long-term engrafting cells, true HSCs, that grow slowly and repopulate the bone marrow over a longer period of time. And so durability comes really once you can confidently predict that you're beyond that first phase of progenitor cells. And for patient number 1 out 28 months, what we're very happy to see is a plateauing of enzyme, whether you're looking at leukocyte or plasma, and an absolute plateauing of vector copy number. So to sum up, very satisfied with the impact on our primary endpoint, kidney substrate biopsy, 30 to 40% further improvement versus ERT baseline, and long-term durability, so far so good. We'll of course track these patients until the end of time, but we're satisfied with what we've seen so far. And in terms of safety, very much within expectations, no AEs or SAEs related to the gene therapy, and the types of AEs that we're seeing are very much in line with the expectation of the concomitant conditioning. So a final point is that really AVRO has been rolling up these lysosomal storage disorder assets, getting them into the clinic, and in the first 8 patients for Fabry disease, we're thrilled with the efficacy that we've seen. But we've actually followed the same playbook as the other lenti companies, which is in parallel to really try to optimize the process. And in 2019, we introduced what we call Plato, which is our commercial stage platform. And Plato really delivers 2 important benefits. Is one, it's optimized for performance. We've tweaked some what we call potency levers to deliver what we hope will be even further confidence that we can reproduce the efficacy that we've seen to date, but also some important process changes that really directly enhances our ability to scale up cost effectively. So in other words, manufacturability. And I'll just show you a quick slide on some of the potency upgrades. Is what, and maybe I'll talk about the changes, is number 1, pivoting to a more state-of-the-art 4-plasmid vector system. Number 2, optimizing our approach to conditioning with therapeutic drug monitoring. And number 3, going to a fully closed automated manufacturing system. And some of the outputs are vector copy number with our academic process versus our Plato optimization, going from a mean of 1 to 2. And importantly, the vector really lets us target whatever VCN we desire, because there really is a strong correlation with multiplicity of infection. So we've decided to target 2 because it just felt better than 1, but still below any range where we would be concerned about even theoretical safety issues. Transduction efficiency, going from a mean of 40 to 70 without any transduction enhancers, and a 2.5-fold increase in enzyme activity with Plato versus our academic platform. And then we really are excited to introduce a therapeutic drug monitoring approach to our conditioning with busulfan, and to essentially take a dose that was given in a one-time bolus, divided into 4, and really target to area under the curve to greatly reduce any concern about out-of-range toxicity. So that's us, and what I would say is we entered 2019 running 2 clinical trials. We exit 2019 running 4 clinical trials, and we're excited to continue to demonstrate the value of the Fabry program and to get Gaucher and cystinosis and then our next generation technologies into the clinic and hopefully show comparably exciting results. Thank you.